Hi, we are here today to discuss developing a clinical research protocol and the logistics to consider as you go through this process. I am Tracy Miller, and I manage the Protocol Navigation Protocol Development Program at the National Institute of Allergy and Infectious Diseases, NIAID. This program consists of protocol navigators and medical writers who assist the intramural NIAID investigators with developing their protocol, starting from study concept through to initial approval by the IRB. As you will learn, there are many things to consider as you develop a study. Here with me is Stacy Kopka, the Senior Managing Medical Writer for the program, and she will start off the talk today going through the basics of writing a clinical research protocol. We are both contractors with Leidos Biomedical Research, Inc., and our affiliation and funding statement appears at the end of this slide. We have no conflicts to disclose.
Every clinical research study needs a written protocol that describes the processes and procedures that will be followed when carrying out the research. It can be thought of as the recipe that will be followed when conducting the study. The protocol is typically a highly structured document that must include specific sections and address a variety of requirements for different oversight bodies, as well as the study sponsor. It is helpful to use a protocol template when one is available, which helps ensure the multiple requirements are met. Regardless of the layout or format of the protocol document, every protocol will include several essential components. The background section provides the explanation of the problem being studied. It does not need to be exhaustive. It should provide a concise explanation of the background, including a summary of studies that have led to the current proposed study. The rationale should provide a compelling reason why the research is important. This often focuses on gaps that exist in the current knowledge base and how the proposed study would help address those gaps and support important advances in the field. Many protocols do not have formal hypotheses, but the idea being tested should always be clear through the background, rationale, and objectives and endpoints. Objectives should be clear statements of each individual goal of the study. For example, assess the safety of experimental drug X. Endpoints usually go hand in hand with objectives and show what will be collected or assessed to address the objective. For example, if the objective is to assess the safety of experimental drug X, an appropriate endpoint would be the number of adverse events and serious adverse events observed following administration of drug X. Every protocol must have a clear description of the study design. This includes an overview of the main statistical design, for example, randomization, blinding, placebo control, crossover design, etc., as well as an overall description of what will happen on the study. For example, how many participants will be enrolled and what their general characteristics are, when and where study visits will happen, how data will be collected, what types of biological samples will be collected, and what types of comparisons will be made. A detailed description of the statistical plan is typically provided in a dedicated statistical analysis section, and a very detailed statistical plan, or SAP, is sometimes outlined in a separate document that can be referenced in the protocol. An important part of the protocol is the description of the study population. This section includes criteria that will be used to narrow down the population of people who will be included in the study by defining the characteristics that will make them eligible and those that will make them ineligible for the study. These are called inclusion and exclusion criteria, and they are typically written as bulleted or numbered lists. The inclusion criteria are criteria that must be met for a person to be eligible. For example, a study that enrolls adults only would list 18 years of age and greater as an inclusion criterion. A person must meet all inclusion criteria listed in a protocol to be enrolled in a study. Conversely, exclusion criteria are criteria that make a person ineligible to enroll. For example, many studies, especially studies of investigational drugs, do not allow pregnant women to participate. So pregnancy would be listed as an exclusion criterion for such a study. A person who meets any single one or more of the exclusion criteria is not eligible and cannot be enrolled in the study. So a person must meet all of the inclusion criteria and none of the exclusion criteria to be enrolled. Another way of looking at it is that as each person is assessed for enrollment, you must be able to answer yes for all of the inclusion criteria and no for all of the exclusion criteria for them to be enrolled. And I'll provide some examples of this during the case study. Any clinical research study that is assessing an investigational agent, such as an experimental drug or vaccine, must have a study intervention section that describes the investigational agent in detail. This section includes details about the formulation, administration, dosing, preparation, and accountability of the study agent. Any dosing adjustments, such as escalation, de-escalation, or other adjustments must also be described. The clinical research protocol should also describe the schedule of activities for each subject that is enrolled. This is often shown as a table or schema that clearly delineates the flow of study procedures completed by each subject from the time they are screened for the study through their last study visit. One of the easiest and clearest ways of showing this information is with a table that lists all of the study procedures and evaluations as rows for each study visit, shown as columns. The protocol should clearly specify when each study visit takes place and provide an allowable window around that time point for the respective procedures to be performed. For example, 1 week plus or minus 2 days after study drug administration. In addition to a description of the schedule of study procedures, each procedure or evaluation should be described as well. A list of procedures that are being performed for research purposes should also be included. Procedures that are not standard or that have specific requirements, such as blood draws at specific time points, should be described in detail. The protocol should describe the risks and benefits of participation. Risks should include any harm that the participants may encounter as a result of participation in the study. This includes any known or anticipated risks, such as side effects from an experimental agent, like an investigational drug or vaccine. It also includes known or anticipated risks of any other study procedures, such as pain from blood draws or radiation from X-rays or imaging scans. Direct benefits that participants may experience should be described as well. Often, participants do not receive direct benefit by participating in research, so this should be stated. Most protocols have a few different aspects of oversight, most commonly safety and ethics oversight, as well as site monitoring. The requirements and processes for each of these will be described in the protocol. These sections are usually very structured and often have a lot of standardized language required by the particular oversight body. Any references that are cited, such as in the background and statistics sections, should also be included in the reference list. It's helpful to use a style guide when writing protocols to help ensure that consistency is maintained throughout the document and enhance readability. The American Medical Association, or AMA, Manual of Style is a thorough style guide that is well suited for the field of clinical research. Next slide. There are several different types of clinical research studies. The specific requirements of each protocol document will be determined by the type of study being described. For example, a clinical trial involves assessment of an investigational drug, biologic, or device. These protocols must include a study intervention section that describes the experimental agent. On the other hand, observational and natural history studies, which may follow standard treatment outcomes or describe the course of disease but do not administer treatments or drugs for research purposes, will not include a study intervention section. Pharmacokinetic and pharmacodynamic studies have very specific requirements that will enable PK and PD assessments and need to be described in detail. Next slide. There are certain regulations that must be followed when designing a research study and preparing a protocol document, depending on whether the study has funding or oversight from the US federal government. These regulations are laid out in the US Code of Federal Regulation, or CFR. Any study that receives funding from the Department of Health and Human Services, or HHS, must follow the federal policy for protection of human subjects outlined in CFR Title 45, Part 46, or 45 CFR 46. Any study of an investigational drug or other product that is regulated by the US Food and Drug Association, or FDA, must follow the regulations found in 21 CFR. There are various parts of 21 CFR that govern different aspects of clinical research, but the parts that typically have the most impact on protocol development are parts 50, which covers protection of human subjects, 56, which covers institutional review boards, and 312, which covers investigational new drug application that must be submitted to the FDA for studies of investigational agents. Next slide. To help put the information I've just reviewed into context, I will present a case example to demonstrate a few aspects of protocol development and the various considerations that go into designing key components of the protocol. The case example involves a fictitious disease and study agent based on an actual protocol we recently developed. I will briefly present the background, study agent information, objectives and endpoints, and study design. I will then walk through the development of participant inclusion and exclusion criteria and an appropriate schedule of procedures. Next slide. This case example involves the investigation of a study drug being developed to eradicate Jumba virus RNA from the body fluids of Jumba virus disease survivors. Briefly, the Jumba virus is a recently identified virus responsible for a recent widespread outbreak of serious, often fatal disease in the southern United States. Although the initial outbreak of Jumba virus disease has ended, local flare-ups occur periodically across the region. Molecular sequencing has been used to link these local disease flare-ups to transmission from survivors. Infection with Jumba virus typically results in severe disease with case fatality rates of up to 75%. The virus is spread through human-to-human transmission by direct contact with contaminated bodily fluids. Data show that the virus persists in some people who have recovered from Jumba virus disease, with viral RNA detected in blood, semen, or saliva for months after the initial disease onset. Recent investigations of disease flares and cluster outbreaks have traced virus transmission to male Jumba virus disease survivors with documented viral RNA in their semen. Thus, safely eradicating Jumba virus RNA from the bodily fluids, such as semen, of survivors may reduce the risk of Jumba virus disease flares and another large-scale outbreak. Next slide. An investigational drug called ST-1017 is being studied in this clinical trial. It is an anti-Jumba treatment that has demonstrated the potential for in vivo clinical efficacy against Jumba virus infection based on preclinical studies. It is administered as an intravenous infusion over 1 hour, once daily for 5 days. Early data from the first ongoing human trials suggest a transient subclinical self-limiting effect on liver function. Next slide. The primary objective of the study is to compare the antiviral activity over 28 days following the administration of 5 days of intravenous ST-1017 versus placebo in male Jumba virus disease survivors with evidence of Jumba virus RNA in their semen. The primary endpoint shows that this objective will be assessed by examination of the assay negativity rate, or ANR, at days 5, 14, 21, and 28. The secondary objectives and endpoints will assess the longer-term antiviral activity and safety of the study drug. Next slide. The study has been designed as a double-blind, randomized, two-arm trial of ST-1017 versus placebo among jumbovirus disease survivors who showed jumbovirus RNA in their semen at screening. Participants will be randomized 1-to-1 to receive either ST-1017 or placebo once daily for 5 days. Antiviral activity and safety will be assessed during short-term and long-term study phases. The primary analysis for the assessment of antiviral activity in the short-term study phase will focus on ANR, which is defined as the percentage of semen samples for which jumbovirus is not detected by polymerase chain reaction, or PCR. Next slide. Now we will go through some exercises that demonstrate the process for developing eligibility criteria and a schedule of procedures for this study. Next slide. As I described previously, the eligibility or inclusion and exclusion criteria are specific criteria that each person must meet to be eligible for enrollment in the study. This section should be constructed to narrow down exactly how the study participants will look based on your study rationale and objectives. For example, if you're studying a treatment for a pediatric disease, your study participants will likely all be children. The criteria will also serve to weed out individuals who may be at increased risk if they participate in the study, either due to risks of the investigational agent or of the study procedures. Next slide. So the questions that we will consider as we develop the eligibility criteria for this study are shown here. First, what are the demographics or characteristics of the target population, or what kind of people in general are appropriate for this study? Perhaps the most obvious clue for answering this question is that the primary objective of the study is to assess the activity of the study drug in males with evidence of viral RNA in their semen. So clearly, we will only be enrolling males into this study. Furthermore, they must have evidence of viral RNA in their semen. We also must consider the appropriate age range for participants. Regulations that govern clinical research outline certain requirements for enrolling children into clinical trials. Children are considered a vulnerable population, so there is a higher bar that must be met to enroll children in clinical trials. The objectives of this study can be met without data from children, so we will restrict our population to adults. Another consideration is whether there is a subgroup of individuals that would be especially vulnerable to risks of the study drug or research procedures. In this case, there are some early data that suggest some possible liver effects. Considering this, we may want to exclude people with a history of liver disease. We should also think about any study requirements that study participants must be able to meet. For this study, thinking about the potential concern for liver effects of the study drug, we may want to require that study participants refrain from alcohol use during and for a period of time after study drug administration. So we could write an inclusion criterion to address this. So let's look at what some of our eligibility criteria might look like. Next slide. As I discussed previously, individuals must meet each inclusion criterion to be eligible for study enrollment. In addition, the criteria should be worded such that the answer to each should be yes when assessing each person's status during the study screening process. So thinking about the considerations we discussed for our study population, our inclusion criteria for this study might include those shown here. To be eligible for participation in this study, an individual must be male, 18 years of age or older, must have a semen sample that is positive for jumbovirus RNA by PCR, and must be willing to abstain from alcohol for study days 0 to 14. As each person is screened for this study, these criteria will be reviewed one by one. If the answer to each is yes, the person is eligible as long as they do not have any exclusionary conditions. So what are our exclusion criteria? Next slide. Exclusion criteria describe the characteristics that will rule someone out from study participation. You must be able to answer no to all exclusion criteria for a person to be eligible. Or in other words, if you answer yes to any one or more of the exclusion criteria, the individual being screened is not eligible and cannot be enrolled in the study. Since our primary objective is to study the effect of the investigational drug in survivors of jumbo virus disease, we would want to exclude anyone with current active disease. Also, thinking about our considerations about potential liver toxicity, we would also want to exclude anyone with evidence of liver disease on physical exam or abnormal liver function tests, as well as individuals with hepatitis infection. In addition, since this drug has not yet undergone extensive testing in humans, we may want to also exclude individuals with other laboratory abnormalities to avoid additional safety risks and also to avoid muddying the safety evaluations that will be done on this study. Next slide. Now we will consider the study schedule and procedures. The schedule of procedures should clearly define what procedures are done for the research and the impact and the exact time points at which they should be performed for each individual subject. The schedule of procedures should be guided by what is needed to address the study objectives and endpoints, while also considering any safety issues based on study risks. We also want to ensure we have sufficient time to complete the procedures required at each time point and that we have the proper facilities and equipment that will be needed. We also do not want to burden participants with excessive requirements beyond what would be needed to address the study objectives. Next slide. So the first question that we will consider as we develop our schedule of procedures is how and when the study drug is administered. In this example, ST-1017 or placebo is administered once daily for 5 days as an intravenous infusion. So we know the first 5 days will require study agent administration and related assessments. Our primary objective is focused on short-term antiviral activity in semen. So we will need to incorporate semen collection for PCR analysis at the specified time points. Our secondary objectives also address long-term antiviral activity and short and long-term safety. So we will incorporate semen collection for PCR at long-term time points and safety assessments throughout the study. Also, we know there are concerns about liver effects. So we will want to be conducting frequent laboratory testing of liver function. So what might a schedule of procedures look like for this study? Next slide. Every protocol must describe the screening window and procedures for assessing the eligibility of each potential participant. The screening procedures should be directly based on the eligibility criteria and designed to allow each criterion to be answered. So for this study, we would need to collect semen for PCR to confirm the presence of viral RNA. We would need to perform a physical exam to rule out physical evidence of liver disease. And we would need to collect a medical history to rule out a history of hepatitis infection, and as well as collect demographic and information such as sex and age. Finally, we would draw blood for laboratory tests to address our remaining exclusion criteria. Once the screening procedures are complete, the results will be reviewed to confirm the individual's eligibility to enroll in the study. Those who are eligible would proceed to the first study visit. Next slide. After enrollment, each participant will start with a baseline visit, which would be considered day 0 of this study. Procedures conducted at this visit should allow the baseline status of the participant to be evaluated. Changes from the baseline status at later study evaluations will allow for assessment of the study objectives. The baseline procedures would include a physical exam and review of medical and medication history, semen collection, and IV catheter insertion to prepare for the study agent administration. Blood would also be collected for the baseline laboratory assessments. Finally, as this is a randomized placebo-controlled trial, each subject will be assigned to a study group according to a randomization scheme. Next slide. Days 1 to 5 of the study are the days that the study agent, either the study drug or placebo, depending on randomization, will be administered. The schedule should clearly indicate which procedures should be done before, during, and after study agent administration as applicable. For this study, the participants' current status, including vital signs and recent medical history, should be reviewed, and the patency of the IV catheter should be checked prior to the study agent infusion. Blood will be drawn for laboratory tests to assess safety and evaluate the study objectives. Next slide. Once the pre-dose procedures are complete, the study agent infusion should be performed. The protocol should indicate any procedures that must take place during the infusion, such as monitoring for adverse events and periodic assessment of vital signs. Next slide. Once the study agent administration is complete, post-dose procedures should be performed. As elsewhere, the protocol should clearly specify the procedures to be performed and any required time points. For example, for this study, vital signs should be collected 30 and 60 minutes after completion of the infusion. Adverse events should be assessed. A targeted physical exam should be performed to assess any signs and symptoms of adverse events, and blood should be collected for appropriate laboratory evaluations. In addition, semen should be collected on day 5 to meet our primary endpoint of ANR at day 5. Next slide. After day 5, participants would return for study visits at days 14, 21, and 28 with plus or minus 1 day windows. The procedures scheduled for these visits should allow collection of specimens and data that will allow us to address the remaining short-term endpoints. Next slide. Participants would return for additional study visits at weeks 8, 16, and 24 with plus or minus 3-day windows. The procedures scheduled for these visits will allow collection of specimens and data that will allow us to address the long-term endpoints. The week 24 visit would be the final visit for this study, and the participant would be completed with study participation after this visit.
So now I'd like to talk to you about the complexity of the protocol logistics. This is very important even when initially designing a study, because it affects how the study will be conducted, and many logistical aspects need to be reflected in the protocol document itself. There are operational activities that need to be considered when you're developing a protocol. As an example, the spokes in this wheel reflect a few of the different offices that may be involved in any one particular study here at NIH. In orange are sponsor responsibilities, such as regulatory affairs, which is the group responsible for communications with and submissions to the FDA. There is the medical monitoring and safety oversight, which could include a DSMB, which is a data and safety monitoring board that we will discuss in the next slide. It is the responsibility of the sponsor to monitor the clinical research site itself, which usually involves visiting the facilities to verify source documents and the data collected and reported, and ensuring that the staff are actually conducting the study as outlined in the protocol. Data management is under the sponsor's purview as well. Here at the NIH, which is made up of several institutes and centers, each institute also has reviewing offices, as does the wider NIH. And whether a protocol needs review by a certain office depends on the procedures in the protocol, such that if it involves radiation safety review may be required. Or if there are biosafety issues with the study agent itself, then it may need biosafety committee review. Of course, all clinical research protocols which involve the engagement of investigators in human subjects research require some level of institutional review board or IRB review. If there will be technology transfer or intellectual property covered under a protocol, then that department would be responsible for assisting the investigator with negotiating agreements with the parties involved. For example, clinical trial agreements or material transfer agreements. Additionally, if products or devices and/or commercial companies are involved, the Institute will conduct a conflict of interest review to ensure that investigators are cleared of conflicts, thereby ensuring the protection of human subjects and the scientific integrity of the research. Even though daunting at times, all of this helps to ensure the proper conduct and oversight of the study. Next slide. Let's dig a little deeper into safety oversight. Obviously very important in any study and critical to think about as you're developing your protocol. The intensity of safety oversight that is needed is somewhat driven by the design of the study agent, and the risk level of the study procedures. Research protocols should include a data and safety monitoring plan that is commensurate with the level of risk of the research to monitor the data collected to ensure the safety of subjects, consistent with regulatory criteria for approval by an IRB. Of course, safety is of prime concern, and safety assessments are conducted throughout the study, which includes assessing for adverse events, which are untoward medical occurrences in a human subject, including any abnormal signs, such as an abnormal physical exam or laboratory finding, symptom or disease temporally associated with the subject's participation in the research, whether or not considered related to the subject's participation in the research. As I mentioned earlier, there are also data and safety monitoring boards. The DSMB is a formal group of independent experts that oversee both subject safety and study performance, which includes reviewing the data and critical efficacy endpoints, assessing the data's validity and integrity, and ensuring that the data is being handled as outlined in the protocol. At NIAID, a DSMB is required for gene therapy or randomized blinded studies, to name a few. There are other types of oversight safety constructs, such as a safety monitoring committee, which may be convened for studies that do not rise to the policy level of needing a DSMB. For example, an open label, unblinded dose escalating design that would benefit from the advice of a smaller, more informal group independent from the study team and with the expertise relevant to the study, whose primary focus is subject safety review. These folks may be able to be convened a little bit more quickly and can provide guidance and advice to the PI at key points throughout the study. There can also be an independent safety monitor, and that's a single individual with relevant expertise whose primary responsibility is to provide independent safety monitoring, especially in studies that might be short in duration, non-randomized open label, or pilot studies. These are just some examples of how safety oversight can be managed. Additionally, the principal investigator and associate investigators and other study personnel who are involved in interacting with the study participants on a daily basis monitor participant safety as part of the routine of research. Next slide. One of the most important issues to tackle and think about when developing a protocol is figuring out how you are going to obtain the information you need on the study. In particular, how will data be captured? Because obviously, how data are collected and ensuring that it is collected accurately is of utmost importance in order to achieve the ability to analyze the data and the objectives that you have outlined in the protocol. So what data are important to answer the research question? What exactly do you need to achieve the endpoint? And then also, what do you need to collect to effectively monitor the safety of the subjects? These are usually some of the most important questions that are pretty common through all clinical research protocols. During development, put thought into the key data points to capture because you don't want to overload trial sites with collecting extraneous data that you may not intend on analyzing. And as Stacy mentioned earlier, you also don't want to burden the participants as well. You want sites to focus on the most important data required and also necessary in order to conduct your evaluations. There are many ways that data can be captured. It can include direct participant questionnaires or by patient report. It can be by medical and research personnel and by direct lab results. All research data needs to be verified with source documentation, which is where the information was first collected. And then there are other things you have to think about. Who else will be checking the data frequently to ensure the quality? And who will be checking to ensure safety of participants as a whole? If you have designed a larger study or one that involves multiple sites, you have to think about the overall safety and who's looking at it from a global perspective. This entails good data management system, keeping track of what is being inputted into the database, and reports that are needed throughout the life cycle of the study to continually check the data. The data management system should be keeping track that the data are being entered correctly and that there are data error checks to make sure that on-site entry is accurate. Such as if a value is out of range and this is happening on multiple participants' records, this could be an early safety signal, or is it a matter of a data entry person missing the decimal point? Additionally, when the data will be captured and recorded in the source documents and subsequently entered into the database, should be outlined either in the protocol or manual of operations. Generally, source documentation happens during the study visit. Then, when data entry should occur, should be outlined, such as it will be done within X number of days after a particular visit, depending on how the visits are structured. Remember, it is also important to consider if the data should be inputted sooner rather than later, especially if this is a first-in-human study, and you want to make sure that data is entered quickly so that you can detect any early problems either within the database or the conduct of the study itself. For example, if the labs are entered after multiple doses have been given and it is found that a certain lab result should have paused a participant from receiving a second dose, this would be a safety concern and a deviation from the protocol. After an incident such as this, the protocol should be reviewed with the staff, and perhaps the data entry should be done more frequently to help prevent this from happening again. The data need to be maintained securely and confidentially in a password-protected secure data system. If data are collected on paper, they need to be maintained in locked files to ensure the privacy and confidentiality of the subject's information. The participant's name and study code should also be in a secure file and accessible to limited key personnel. Case report forms, or CRFs, can be paper or electronic. Once entered electronically, the data can be cleaned efficiently with queries. Ongoing quality control checks are also essential. Next slide. Okay, so just with the brief overview, I wanted to refer back to the case example and ask you all to consider what safety oversight and data capture would be needed, or what would you want to see if this were your study based on the study design? What additional oversight is needed according to what I've just discussed with you? A DSMB would be needed because it's a double-blind randomized study. The data will be blinded to the participants and investigators. The DSMB can unblind the data if they suspect a safety concern. Next question, what, how, and when should data be collected? Answers are based on the protocol's schedule of evaluations. You would want to make sure that the eligibility is checked before proceeding with any research interventions. You would want to have safety labs, the results of physical exams and vital signs, and the study agent administration collected in the data capture system. And a CRF to collect descriptions of adverse events and serious adverse events, which would also include the start and end dates of these events, the toxicity grades, and the relatedness to the study interventions as assessed by a qualified investigator. How relatedness is assessed is outlined in the protocol, as is the toxicity grading scale. Since infusions for this case example are happening 5 days in a row, you may consider having the data entered daily during this time, and labs and any adverse events should be reviewed prior to the next day's dose. Whether electronic versus paper CR system is appropriate depends on the data management system being used, where the entry will be done, which includes the feasibility of data entry on site, and access to source documents to verify the research entries. If the study takes place in a remote setting where access to either an electronic system or the internet is needed, and the researchers are out in the field, it might be necessary to use paper CRFs and record observations on source documents, and then take that information back to the data management center to enter the data onto a CRF or into an electronic system. Decisions like these are made depending on the resources available, which include site personnel and location of the trial sites. These are things to consider when designing a study and thinking about operational logistics and feasibility. In this case example, since the study agent needs to be administered via IV, there needs to be a medically equipped clinical trial site available, and therefore likely that electronic entry could happen there. Okay, so if safety is an endpoint, how would you quickly assess safety? For example, what results do you need prior to the infusion, and how will they be reviewed to ensure the participant should be dosed? There would need to be a plan described in the protocol for the lab result reviews pre and post dose. For example, perhaps ensuring the liver function tests are within the expected ranges, since the study agent is known to cause mild elevations in these tests. A responsible study member would then review and make a decision about dosing and safety to proceed. There would also be a delegation log separate from the protocol itself that clearly delineates who is doing what and their responsibilities for each member of the study team based on expertise. This would also be discussed at the study initiation visit with the site monitors. Then last but not least, once the data are captured in whichever system you have chosen, you have to ensure that the plan is working and that data are being captured efficiently and accurately. As a study investigator, it is your responsibility to ensure that the study team is doing their job, and there should be checks in the system and in the site operations themselves to make sure you are sticking to the protocol specifications. In this case, since infusion happens 5 days in a row, there should be sufficient quality checks during that time to ensure adverse events are assessed quickly. Next slide. The next thing to talk about are some of the other logistics, such as determining who will be the sponsor of the study. This could be based on several external factors, such as where is the funding coming from? Is it through a grant or a pharmaceutical or biotech company? Is there a network or consortium of collaborators? What resources are available to the principal investigator from various agreements that may be in place? And where is the study actually happening? Once a sponsor is determined, then they generally are the folks that prepare the investigational new drug application for the FDA if it's a regulated study. As we've discussed earlier, they will also be responsible for the safety oversight and the site monitoring. These topics are also reflected within the protocol document itself in various sections. The different reviewing bodies will also look at these details to ensure there is proper oversight. The sponsor also needs to review and approve the content of the protocol and consent. There are also more logistics involved for multi-site or international studies. Therefore, if it is determined that the study design should include multiple sites or has to be conducted overseas, then additional IRBs or ethics committees will need to review the study. How this fits in with your timelines and budget needs to be taken into consideration. Additionally, documents may need to be translated, and operational planning in each country or at each additional site obviously increases the level of complexity and management that would be needed. But it can also facilitate your recruitment and enrollment of the study, especially if it's a time-sensitive issue or if the particular topic of the protocol is affecting multiple areas or regions. It might be most beneficial to conduct the study in those regions. Next slide. Based on the case example, if you're the PI of this study, what should you consider? Should this be a multi-site study? Perhaps, but it could depend on the sample size and ability to accrue subjects or on the primary site's capacity to enroll. If you're looking at your home institution, how many patients would be available given the eligibility criteria and design of the study, and what you know of your institution and patients? Are there collaborators with whom you've discussed the study and who may have identified potential participants to refer to your site? Or is it better logistically for those collaborators to be added as sites? You also have to investigate where the disease is predominantly occurring. Is it regional, seasonal, or more global? You may need to go to where the outbreak is happening, as opposed to having participants come to you. So the location of the potential subjects is important, and the interest and expertise in conducting the research of this nature cannot be overlooked. There might be other sites that have investigators who really do want to support you in this endeavor and are interested in doing this and have the resources and expertise to do a study of this type. For this case example, you would need to target sites in the southern US, in the regions where the outbreak and flare-ups are occurring. Thinking about who will be the sponsor. In this case, it seems Scientific Treatment Inc. could do it as the maker of the study product. They can also contract out sponsor responsibilities if they do not have certain capabilities. Now, how many institutional review boards do you think you will need to review and approve this study? It really depends and is based on the number of sites and where they are located. If there are multiple US domestic sites, as may be the case in this example, and it is NIH funded, then all sites would fall under one central IRB per the regulations. If it includes international sites, most likely there would be an in-country or local IRB or ethics committee as well. As Stacy mentioned previously, what regulations are followed depends on the study. In this case, both US and FDA regulations apply, and if it were to be conducted overseas, then additional local regulations would also need to be followed. Next slide. Here are some additional references for your consideration. And last slide. Thank you everybody for your attention, and good luck if you pursue clinical research and protocol development. It's a fascinating and rewarding field. Thank you.